to start with this, remember that we will be talking about the virus in prokaryotes. Okay, so the first thing that I have to share with you is that the viruses, well, are small non-living things which cannot replicate outside of a host cell. A virus consists of genetic information that can be DNA or RNA code with a protein. Okay, so you'll have that thing like an envelope around the DNA or the RNA that will be like protecting the information of the virus. Okay, and this is the way that the virus can attack. Let's say a virus injects its genetic information to a host cell and then takes control of the machinery of the cell. This process enables the virus to make copies of the DNA or RNA and make the viral proteins inside of the host cell. The virus can quickly make multiple copies of itself in one cell, release these copies into to infect new host cells and make even more copies. In this way, the virus can replicate very quickly inside a host. Okay, so you can read this if you want, but I want to go right through the images. Okay, so these are DNA and RNA virus. I already mentioned one of the types of RNA virus. We have two types of RNA virus that I want you to know. One of them is this one, and the other is the one in the bottom that I already told you about it. This is how a DNA virus can attack to a cell. Okay. So it is by steps. Okay. So the first step here. Okay. It says the virus enters the cell and is encoded, releasing the viral DNA and the capsid proteins. Okay. First of all, I have to tell you what is a capsid. Okay. This is where you have the virus parts. Okay. The DNA is the unique material, and the capsid is like the membrane of the virus. Okay, but it's not a membrane. It's called capsid. It's just like proteins around it. And then, okay. So you have the DNA capsid. Okay. So the step number 2 right here, it says host enzymes replicate the viral genome. So as you can see here, first of this, blue. Okay. It says this is the host cell. So the viral DNA will be like this one. Okay. That viral DNA will be replicated by the cell here. Okay. This is like the replication of the viral DNA. And there is another thing. Besides that you have the replication of the viral DNA, you also have the transcription. This is where you have the transcription. It says this. Meanwhile, host enzymes transcribe the viral genome into viral mRNA, which other host enzymes use to make more viral proteins. Is this okay? Let's suppose that you have like so. Again, from the viral DNA you got the mRNA, and from the mRNA you got the proteins. Those proteins will make another structure of the virus, because these proteins get together with the viral DNA that was here. Okay. So the viral DNA from here and the proteins from here will assemble a new structure of the virus right here. Okay, it says viral genomes and capsid proteins self-assemble into new virus particles, which exit the cell. Okay, so that's like the last step in a DNA virus. So this is the RNA virus. An RNA virus, guys, is a little bit different because you got RNA instead of DNA, okay, but you still have the capsid. Sometimes the capsid is like this. It says envelope with glycoproteins. This means that it has something like a membrane, but it's not a membrane, okay? I don't know. So it says glycoproteins on the viral envelope bind to specific receptors molecules, not shown here in the figure, on the host cells, promoting the viral entry into the cell. Okay? So the virus enters the cell, and then the capsid and viral genome enter the cell. Digestion of the capsid by cellular enzymes release the viral genome. So you already have the viral genome. So, but in this case, it's not DNA; it's RNA. So that means that you have like a step less, okay? Because the cell doesn't need to transcribe the viral DNA because you don't have DNA, you have RNA already. So from the mRNA, that is this one, you'll get copies of the mRNA for the new viruses, and you'll get also this mRNA that will help the cell, will tell the cell, let's say, because it's not helping, how to make the capsid proteins, how to make the glycoproteins, okay, well, the glycoproteins right here, and will also tell the cell how to got everything together again, okay? The glycoproteins with the new virus particle right here, and you'll have new virus particles. Okay, you see each new virus spots from the cell. Its envelope starts with viral glycoproteins embedded in membrane derived from the endoplasmic reticulum. Okay, so this is the way that an RNA virus can act inside of the cell, or can replicate inside of the cell. So it's a little bit different here than the other one that I already showed you here. Okay, so these are DNA and RNA. There is another one of RNA viruses that is called retroviruses. Okay, the retroviruses are those ones in which from the RNA of the virus, you have to do, the cell has to do a process that is called reverse transcription. Okay, so you got first from the RNA of the virus, after a reverse transcription, DNA, and from and then that DNA has to go through a process that is called integration. That is that it inserts into the DNA of the host, and then after a transcription, you got the mRNAs, and after the translation, you'll have the new proteins, the new viral proteins, and finally you'll get new viral structures, okay, new viruses there, okay, like here. This is the one that we talked about the last class, okay, that is a retrovirus. These are DNA and RNA viruses that are not retroviruses, okay. Now these are some types of the virus particles, okay. This is the tobacco mosaic virus, okay. This is the adenovirus, okay, the influenza virus, and the bacteriophages, okay. So as you can see, we can have different structures for the virus, okay. These are the ones, these ones look like cones, not like cylinders, okay. These ones have like strange, like a polygon figure, I don't know, okay. These ones look like a cell, but with proteins in the surface, and these other ones look like spiders, right? So the thing here is that, at least for the bacteriophages, okay, the bacteriophages can attack bacteria, okay, that's why they are called like bacteriophages, T4 in this case. So each of these ones have the DNA or the RNA inside of the capsid. So in this case, the head is the capsid, okay. In this case, the capsid is this one, okay. So you have the RNA inside, the DNA inside of these ones, and the RNA inside of the mosaic, inside of the cylinder shapes. Okay, and they look like that. These are images of electronic microscopy. Sorry. And in those images, you can see that they have the shape that we can have here. Okay? And then, all the viruses, well, like the mission of the viruses is to cause an infection. Okay, so we'll have common diseases. Okay, so this is the activity. This is the activity that I want you to look for right now. It says, look for the name of the virus responsible to cause the following diseases. You got hepatitis, you got the AIDS, you got the common cold, you got influenza, you got chickenpox, zoster, and rabies. Okay?